watch this recording, how would they do that?
I will be sending out a link. Helen's
And is that then on PanCAN's website if, say, someone from Seattle or Chicago or some other setting would want to watch this?
That is going to be on our Facebook page.
Okay.
Okay. So welcome, Dr. Jonathan Brody. Thank you for joining us this evening. I'm Stephanie Ross. I'm the Survivor Outreach Chair for the Portland affiliate of PanCAN, and I'm joined by Sue Best, my co-facilitator, who is a palliative care social worker, as well as having other connections with pancreatic cancer. Dr. Brody, last time you were here with us was a little bit over 2 years ago. And you told us about what you were doing in the lab, but you also told us that speaking with us inspired you to go back into the lab and take a look at something. So I was wondering, have you pursued that? What's going on? Tell us all the new stuff.
Yeah, sure. So I was wondering, I feel like I still feel like we were sort of battling with COVID.
Yes, it was October
at that time.
2022.
Yeah.
Yeah.
And I'm really honored to speak to be asked to speak here. And I apologize. I think last time I had a conflict, but since then some of my colleagues have spoken. And to your point about medical advice, I mean, again, Dr. Shepard is number one. So the fact that you've all been touched by him or understand him, we have with him and Dr. Lopez and others at OHSU, we really have an amazing group of clinicians and clinician scientists and scientists like myself who are working on this problem. By way of sort of your question, Stephanie, I don't know if you want it to be this formal, but I did put together some slides.
Okay.
I can share them if people want,
Sure.
just to sort of, so let's see if I can.
And while you're setting that up, why don't you tell us your why? Why did you get into pancreatic cancer research?
Oh, I don't want to take up the whole hour.
Okay.
No, I'm kidding. I'm kidding. So, really, I'm a scientist at heart, and I did my training, and I'll answer the question. I'll kind of also introduce myself a little bit. Is that I did my training at Johns Hopkins, and at the time that I was at Johns Hopkins in Baltimore, it was really a golden era of our understanding of the genetic basis, if you will, of pancreatic cancer. And so when I got my PhD there, I worked in a laboratory of Dr. Scott Kern, who described more mutations, genetic mutations, so alterations in DNA in pancreatic cancer, more than anyone else in the world. And so it was a very exciting time. But even though we knew the genetic code, if you will, of pancreatic cancer, we still needed to do better, and we needed to crack that code. And at that time, and it sounded like maybe Sue had mentioned this, might have this subtype of the disease, which is maybe why you're doing so well. We found that there was a subset of the pie. If you think about pancreatic cancer as, all of it is a pie, that there's a pie chart, and that there's certain subtypes and brands of the pancreatic cancer that should fit a specific and be specifically sensitive to a specific cocktail of chemotherapy or drugs. And we're actually at a really exciting time with that. But at the time, we had sort of made this breakthrough connection based on other people's work and our own, that there were certain brands, if you will, of pancreatic cancer that could be sensitive to this specific type of therapy called PARP inhibitors, or part of FOLFIRINOX, which is the oxaliplatin, the DNA damaging agent. So as I was training for my PhD and went through my postdoc, there was another very talented surgeon, like Dr. Shepard. His name's Dr. Yo, who came from Hopkins. And he recruited me up to another institution and said, hey, look, we need to really have other institutions who are really studying this disease, because this is a really difficult challenge. And so he recruited me up from Baltimore to Philadelphia to sort of lead this pancreatic center. And so I was there for a dozen years working on this problem, and we made some discoveries, but it was really until Dr. Shepard and Dr. Sears, who co-direct the Brendan Colson Center here at OHSU, they recruited me out here in 2020. Um, and so since then, I've just been really, to be honest with you, fascinated by the problem, but also along the way would have this, and I'll show you this in some slides. Um, we run a patient symposium at Jefferson, and so I've made a lot of connections with a lot of patients and their families, and realized that that most of the people that I had met, just like me or anyone else, healthy people, and as we know, cancer and specifically pancreatic cancer randomly picks its quote unquote victims that they want to harass them and their family members. And so I feel like we needed to do something better. And so I'm partly, would like to do good in the world, and made a lot of connections with a lot of survivors. So want to help. And then in part, intellectually, I really want to, kind of be part of someone who can kind of help put the puzzle together so we can really figure this thing out. And so there's a lot of great people who are working on this, not just my lab and the people in my lab, but I think those connections, as well as the intellectual connections, really kind of allowed me to focus specifically on this disease. And so these are the people in my lab who do the majority of the work. My son in the middle there actually, although I try to put a pipette man in his hand, he's not really ready for prime time yet. But this is the part of our group. And back to this is the patient symposium that we did at Jefferson, and we're actually I'll show a flyer. We plan on doing this at OHSU in April, and so I'll put a flyer out, and you can reach out to me. Dr. Shepard or anyone else. And I think Dr. Zimmer is probably, I think, spoke to this group she had mentioned.
Yep.
And she's really kind of really running with that. But this is an interesting, question. We asked the question to all the patient survivors as well as, and you're all doing quite well. But we asked the question of the patient survivors. We asked to them and their families. Avi Nevler is a very talented assistant professor who was at Jefferson, who was in my lab. He asked a polling of all these folks and said, "Okay, we give these statistics, and you all see the statistics, and you can go on Google and see what the statistics are for pancreatic cancer. But what do you think is a cure? What makes the difference for you?" And basically what he found is that the number one answer that, and it'd be interesting to hear what you all think, but the number one answer that patients and their caregivers want to know is, they say, well, if you want to call a cure and make a difference in this disease, I want to live as long as my age match peer. And so this, we don't want to go for 5 years. If I'm diagnosed at the age of 60, I want to live as long, to 85, 90. And so it turns out that we're really kind of, if you look at the cure rate based on what I'm thinking you all want and what you all deserve, we need to do better. And these are old data, and we actually are doing better. And so based on that, we want to set up, this is a little flyer put together for you all today. It's a save the date. I would say it's 90% going to happen. But this is April 19th. It's going to be a free conference where we're going to give education and celebrate you all. And this is going to be hopefully some information that's useful, like nutrition. What are some of the new potential exciting discoveries and therapies that are out there? So more to come on this, but we plan to do this Saturday, April 19th. I believe we're planning on doing this on the waterfront. And of course, Stephanie, we will give you the information so you can disseminate this
Yeah, I know
as you.
Helen and Jen Maldonado are aware of it, and
Okay.
we're all about helping you in whatever way we can on behalf of PanCAN. So
Thank you.
you're welcome.
So, just to give you a brief view, and I think some of these slides I'll go through fast because as always, I probably put too much stuff on, but I really think some of these things are what we're going to make a difference with not just at OHSU, but across the country, and then with this disease. And I think a big thing is you all are obviously, I feel like you're sophisticated, educated people who are getting the best of care. But it actually turns out, which is really, some of the statistics are really bad, that, for instance, there's some people who get diagnosed late, and because of that, and because their caregiver in the community doesn't understand pancreatic cancer, they don't even get offered surgery when they could have been offered surgery. They sometimes get offered chemotherapy late. This is something that we can make an easy impact on, and I'll sort of talk about some of that work we're doing in the state. I think we need to bring young folks into the field. We really need some bright minds to go into this. I think because of, unfortunately, the sobering funding aspects from NIH and everything else, a lot of young investigators don't want to go into this field, but we're doing a good job of doing that, and we're actually doing a pretty good job of that at my institution here. We understand this is a very complex tumor, and when people talk about a tumor microenvironment, we realize that we don't just have to focus on the tumor cells, but we have to focus around the cells around the tumor. And sometimes, for instance, that makes it difficult for drugs to penetrate to the tumor cells. And we also have to think about the immune environment that's going on. And we don't have a great understanding of why primary pancreatic tumor cells metastasize to the liver and lung, specifically. Although we just got a publication out of the Sears lab that talks a little bit about that. And then some of the exciting stuff that I think is really hitting the pavement now, and probably be ready for real prime time in about a year or maybe less, are these KRAS inhibitor drugs. And I'll talk a little bit about those. You might have heard about them. But first, some work that we're doing in the state of Oregon, I'll quickly go through this. This is just a little bit more of some outreach stuff. It's really interesting that it turns out that that in Indian country here, specifically, we're looking at Warm Springs tribes and the Klamath Falls. There seems to be a twofold increase in the rate of pancreatic cancer. And there could be a lot of factors for that. There could be modifiable factors. We don't know. It could be because of type 2 diabetes, obesity, tobacco use. But the thing that's really kind of shameful from the OHSU front is that pancreatic cancer patients in our Knight Cancer Institute are probably one of the #1 accruing patients to clinical trials, which is really important, and shows what a focus we have on pancreatic cancer at OHSU and the Knight through the Brendan Colson Center. But I think in the past, it's something, and I'm not quoting myself on this because it's something, it's either worse or better, but it's like in the last decade, we've had one Native American patient, even though there's such an increase risk there. So with Dr. Shepard, who has a big interest in this as well, and Dr. Shan and Dr. Clay Killsfirst, really interesting, right? So Dr. Clay Killsfirst is a PharmD who treats patients with pancreatic cancer. He's kind of in charge of the drugs. Dr. Shepard, we've talked a lot about. Dr. Shannon is our outreach specialist at the Knight. We all came together and we decided we need to do something about it. So we looked into what are these potential causes. So one is late detection. Sometimes on the reservation and otherwise, these people may not recognize that they have symptoms. I'm I'm certain that all of you have your stories of thinking what the symptom was. And a lot of the time, unfortunately, like people go and you go to your internist and they say, "Oh, it's back pain." Well, we're all of age where it's like, I know for me, if I pick the vacuum up the wrong way, right, I could say, "Oh yeah, that's because of the back pain." And so we all kind of, get upset because we think about, well, what if the doctor really listened to me? Um, but I mean, in a lot of cases, we know when the symptoms really come on, and that big differential probably doesn't make a difference. But you can imagine if someone had gone back to the doctor maybe 5, 6 times over the course of 2 years, and they're constantly the physician never had experience with that. Um, and so lack of access to care, there's a lot to do with, as probably you all know, with historical trauma, lack of trust, and then there's basically zero research that's being utilized here. So with Dr. Claes Kilfirst, again, he's another young investigator that we brought into pancreatic cancer. We've actually gone out to the Confederate Tribe of Warm Springs, and we've done these sessions with the Health and Welfare Committee. And we've answered questions to folks about symptoms, about research. We've gotten a Department of Defense grant. We've also gotten a local CUNY Foundation grant. Along the lines, I'm part of this project called the Healthy Oregon Project. You could all look it up. You could all sign up for free if you're interested. And basically, these are the amount of Oregonians that we have touched in blue, who have actually, what you do is you can Google it, sign up, and we'll send you a kit, and you send your saliva back. We sequence 32 genes, and we sort of tell you if you're predisposed to cancer. So, or potentially other cancers, or, what it means as far as your germline's concerned. And the red dots are people that we've identified. And so the goal there is that we want to actually set up a specific, what we call POP, which is a Pancreas Oregon project, and try to figure out ways using electronic medical health records, as well as look at high-risk areas, as well as their genetics, and basically find out patterns. Can we predict up front what areas and what people might be high-risk individuals going forward? So what do we know about the molecular aspects of pancreatic cancer? And so this schematic here that I have set aside, let's see if I can you see my cursor?
Yeah.
Okay.
Yeah.
So this is from a normal pancreas all the way to pancreatic ductal adenocarcinoma. And these are the PanCAN lesions that everyone has who doesn't even, who may not even have pancreatic cancer. Or there's some studies that have come out that a lot of people have precursor lesions. I'll get into the point that we're studying a little bit of, like, why do some people convert from these precursor lesions to pancreatic cancer? And below are the different tumor microenvironment cells that sort of support the pancreatic cancer. And so there's a number of things we know about this progression model, if you will, from normal to pancreatic ductal adenocarcinoma, or PDAC. We know that there's a number of these mutations in these specific genes that cause that. See if it allows me to move forward here. So why I'm trying to figure out why this isn't moving forward. Let me stop and ask any questions. I know I'm throwing a lot of information at everyone. Anything that anyone specifically wants to ask, or should I just keep moving?
You can keep moving. I want to let everybody know that I am in that Healthy Oregon project, and it was very easy. And every once in a while, you get a little notice that you have some questionnaires to fill out. And once you fill out the longer family history one, it's just pretty easy. It just takes a few minutes the first time. So I highly recommend it.
Thank you, Stephanie, for saying that and doing it, and being one of the 50,000 Oregonians that have stepped up. So we appreciate that. Okay, so this is a protein. So, you might have a lot of scientists speak to you, and the pictures of the people are the people who've done the work in some of our publications. But we study this protein that's highly active within the majority of pancreatic cancer cells. People call it HUR, HUR, HER. So we utilize this protein to do different things, to study, find new targets, study how the immune system avoids pancreatic cancer, how it is involved in development of the microenvironment, how it drives metastasis, and then also how it's involved in this molecule that you might have heard of called KRAS and KRAS mutations, which are in basically 95%. So this is how, like, when we look from a pathology point of view, this is, a very colorful image on the right here of pancreatic cancer. And this is the staining of our active protein HUR in green. And you can see how active and ubiquitous it is. And so it turns out that when you look at tumors on the left side and you stain in brown, you can see that this protein is highly active in pancreatic cancer. And so here's a depiction of that progression model again about how in light, the normal pancreas doesn't have a lot of the protein as depicted in brown. But if you go to the right, you can see that it increases its expression. And this is just a schematic of that. And so, really there's a belief that there's a lot of hope on the way with these KRAS inhibitors. Have most of you heard about these KRAS inhibitors?
Yeah.
Okay.
Yeah.
And, for some, you're all doing well. And at some point, when these become ready for prime time, there is a chance that your tumor may see these, if you do have tumor cells that reside, they might see this therapy. And this is probably one of the most exciting precision oncology therapies that are out there, because this precisely goes after a mutation that's should be only in pancreatic cancer and not in your normal cells. And so this is a specific lock. If you think about a lock, this is a specific key that will go in and block that lock. And that lock is key to basically, the survival of pancreatic cancer cells. And so I'll show you here, how exciting this drug can be is that you can see, you can see here, these are stained pancreatic cancer cells in a dish in the lab. And if we just add small amounts of this drug, literally within about 24 to 48 hours, these cells completely die. Now, we think this protein that we study, HUR, is critical because if you remove HUR, genetically engineer HUR out of these cells, you could see that the cells survive. They become resistant. And so what we think we're understanding is this concept of resistance, and we show that we can do this in animal models. What's important about this is we know that we can really do a nice job with this, but there does seem to be some pancreatic cancer cells in some people in some of these early trials that seem to become what we call resistant to the therapy. And if we can understand the resistance, then we could play chess, if you will, with the cancer cells so we could stay ahead of the game. And so that's what Hen Alamesh, who's a talented postdoc in my lab, is doing. This just shows again that these are pancreatic cancer cells that were basically, and this is a survival curve, that as we increase the amount of KRAS inhibitor, we're basically killing every single pancreatic cancer cells with these KRAS inhibitors. This is data that we haven't published, but we also show that if we take this HUR protein away, by genetically modifying these cells, they become resistant to the therapy. And this just shows, what happens in the animal model, that when we treat, we lose this HUR protein after we treat. So I'll probably run through some of this data, but what is really exciting is that you can see here that pancreatic cancer cells have this in green, this HUR protein is very lively, it's very active, it's protecting itself from any sort of damage. However, if we add this KRAS inhibitor, you can see that it basically goes away. And so these cells are on their way to dying.
So can I ask you, what do you mean by low glucose stress?
Great question, Stephanie. It leads into this slide.
Okay.
So it turns out that pancreatic cancers and their cells, they live in an environment that is nutrient deprived. They're basically starved. They don't have a lot of access to a lot of food. And it turns out that decrease in glucose or their ability to, or even oxygen, puts them in a very strong setting. My colleague likens it, Dr. Winter, who's at Case Western, he always uses the analogy that it's like basically it's a cactus growing in the desert. That you can imagine, we often think of a cactus, it's growing in a very nutrient deprived environment. But I don't know about you, but I often think of cactus as being pretty sturdy, right? They're pretty strong and they can survive a lot. And so that's what happens a little bit with pancreatic cancer, is that we're up against these kind of these cacti, if you will, that can store energy and really kind of survive. And so they actually become resistant to therapy because of that. And so this study that we had done here is that if we induce increased glucose availability to pancreatic cancer cells, and we look at that both at patients and in mice, we can actually improve FOLFIRINOX. So for instance, if you just look here, in an animal model, you could see that FOLFIRINOX, works pretty well. This is the size of the tumor. So as it goes up, the tumor goes bigger. However, if we give the animals sugar water by basically inducing diabetes, the tumors shrink. And that's just because we're able to block that. And so what we're trying to do is in the lab, what Tobias's doing is basically if she increases the amount of glucose, and so we basically add 25 millimolar glucose to the pancreatic cancer cells, we see that the KRAS inhibitor works really well. There's no surviving cells here. And so one of the grants that I have in with this gentleman, Dr. Winter, is we have a clinical trial that we're writing up where we're trying to allow permissive hyperglycemia or induce diabetes while we're giving chemotherapy to see if that will systemically improve therapy. So we've published this idea, and now we're trying to get money from the NIH to actually run the clinical trial. And so we want to do the same thing with the KRAS inhibitor. So it's a little bit out of the box. And I don't know, you could tell me, we're not sure patients would really like the idea of doing it, but it would just be a short-term permissive diabetic situation.
And that is the hope then that after the chemo is over, that you would revert to being not diabetic?
Correct.
And
Correct.
so, and the other question is, are we eating the birthday cake or not?
Yeah.
I mean, laugh because the only thing I could eat on chemo, because I was by myself, my mainstay was Honey Nut Cheerios, which was full of sugar. And everybody said, oh, the worst thing ever. But here I am.
Yeah.
I mean, I wouldn't say it, and I'll speak for him because he's one of my best friends. But Dr. Winter here, if he was on, he would probably say, well, that may be why he had such a good success.
Thanks.
But
so
no,
Julie developed diabetes early in this trial, so she's doing better than anybody else. So go diabetes, it's good for you.
Yeah, there you go. Yeah, and I'll go back to Stephanie's comment. You do not want to take any medical advice from me on that. But what I was going to say, look, I understand. My mom is currently, she's got stage 3 endometrial cancer right now. And she's on her third cycle of chemo. She's 82 years old. And it's really interesting that well, as a side thing, it was kind of interesting because she has, different things that she feels like she could only eat when she's going through her cycle. But, she called me the other day, and I'm just relating this kind of it's, whenever you talk about your mother having cancer, it's never a funny story, but this was sort of a funny story. Was that she called me, she said, Jonathan, well, she runs everything by me. And what's really interesting, she has a molecular marker, so she's specifically going on a Herceptin, HER2 positive therapy. So knock on wood, hopefully she'll be okay. But two things I want to sort of relay to this group about that story. One is she called me, she said, I was on this Facebook group and they are saying that one way that I could manage my side effects is if I do like some sort of intermittent fasting and I don't eat before I go to chemotherapy. And this is the opposite of what I'm saying here,
Yeah.
right? So she said, "What do you think about that?" And I said, "Well, Mom, we published this paper." And it doesn't necessarily agree with that. And I said, first of all, that doesn't really, I don't know, maybe like if you're so hungry, you just think about your hunger that you're not, the science behind it didn't make sense. So anyway, luckily she called me because she was about to fast like 12 hours before she was going to get her chemo. So I'm glad. On sort of a sobering point, it really hits home. My mom's been getting therapy now, for a month, and she's going to get it through March, right? And we don't, she's actually going to get her PET scan tomorrow, and I'm flying home actually tomorrow, to be with her. And the thing is, it's amazing that hits home to me that you all know this better than me is it's amazing that like we go through this intense therapy and it's with like the hope that we're going to get a response, but we're not going to get that data for so long, right? It's not like a blood test where you might have to wait a day to get that, but to go through, you know, I've sat with her now a couple of times while she's getting her therapy. And it's intense. It's an emotional thing, and I use this tangent because I think one of the things that I'm not really going to talk about that I think is really important is the future of being able to monitor people better and faster of whether the therapy works or not. And there's these things called CTCs or ctDNA where we could actually, through blood work, we could find out pretty quickly if someone's responding. And it's more specific than things like CA19-9, which I know you all know about, and CA125 and everything like that. So anyway, this is here to say that obviously it wasn't in that, but I really appreciate more than ever, seeing my mom go through the chemotherapy and her calling me up every morning and saying, is it going to work? And I wish, to whoever you believe in that that it's going to work, but the fact that she is 2 or 3 days after chemo hearing that she's just devastated, can't get out of bed, and knowing like what is that worth it is it really makes me believe that we need to continue to work harder and harder in the lab and continue to do these things. So sorry, I didn't mean to go on a side.
Yeah, I'm so sorry to hear that. But I'm glad she's able to receive treatment and that you can be with her.
So thank you for saying that. And she has that attitude, she has the attitude of she's grateful that she's getting therapy. So I just hope it's, we don't, yeah. So hopefully good news there, but we'll see what happens. But thank you for allowing me to share. So I think from this work, we're going to figure out what these resistance mechanisms are. This is just kind of a busy slide, but basically what this says is that there's a huge part of my lab right now is trying to stay on top of these KRAS inhibitors so that when they become ready for common use and standard of care, they might get to a point that, for instance, in a maintenance setting that, Judy might be able to get this therapy just to make sure that there's no cancer cells left behind, or any of you at any different points. So we want to make sure that we're optimizing this as best as possible.
Is this
So
pan KRAS all the alleles, or which each one is individual?
Great question. So it's everything. So this is not just the specific G12D one, but we've also done this with the pan-RAS inhibitor.
Okay.
So yeah, and the therapies would be against primarily the majority of like every KRAS inhibitor that's out there.
Thank you.
Yeah, thanks for the question. This slide really just shows the complexity of trying to target the immune compartment of pancreatic cancer. These are the different molecules that we look at. This is from a review that we just published in Journal of Clinical Oncology Advances. But what I want to show you here, I'm going to kind of cut to it. This is amazing. If we basically block this HUR protein, these pancreatic cancers are 100% rejected by the host, this being the mouse. And what we found is that this HUR protein causes this immune suppressive environment. And we're also finding, that some of these immune oncology agents that you might have heard about that work so well in like melanoma, certain types of lung cancer, et cetera, they don't work so well in pancreatic cancer. And we think we're figuring out why there's some resistance. And we think it's because, again, of this protein that we're studying. And so here, when we use this PD-1 therapy up top here in a mouse, and we just silence this HUR protein, we can see that we are able to basically deplete the size of the tumor by just knocking that protein out. So this is a huge part of my lab doing that. I'm going to skip over this and tell this one exciting story that Rick Burkhart with Jordan Winder, we basically figured out that one of the proteins that HUR regulates is a metabolic protein. And we think that this metabolic protein is really important for keeping pancreatic cancer cells alive. And it turns out that there is an inhibitor that is available against the mutation. However, that mutation doesn't exist in pancreatic cancer, but what does exist is these low magnesium levels, which will allow the drug to work. And so after 10 years of work here, we've basically been able to figure out that this drug works specifically in pancreatic cancer cells. And we've set this up as a clinical trial with Case Western. And so this is just to show that, 10 years of work later, we finally got this drug into the clinical trial. And that's why, again, I'm so excited about the KRAS inhibitor work.
Very cool.
So I'll end with, I want to give a little time if we want to have a discussion. And I realize people, how much time we spend here. But this was a PanCAN funded study that I was part of, where we basically went in 50 states, and it's called the Know Your Tumor Program. Some of you might have heard about it. I'm sure Stephanie has.
Yeah.
Where we went and sequenced everyone's tumor, and we made it, basically, we democratized personalized medicine. And it was a way of not just educating the patient, but also the clinicians that may have thought about this. And what we found is that patients that actually had mutations that matched a therapy lived a year longer than patients who didn't. And these were all metastatic patients who, really didn't have good prognosis from the beginning. So that's why these numbers are so poor. However, we could see that there's a huge difference in the lifespan of the people that we've done. And now I think it's way over 10,000 patients that we've sequenced on this program. So, the big vision here, and this is with Dr. Sears here, is we're trying to set this up of trying to figure out in a very sophisticated manner, and we're doing this with Johns Hopkins and the University of Maryland, is we're using all these fancy different assays to really figure out why certain people who have precursor lesions go on to pancreatic cancer. And so that's this before pancreatic cancer research center activity we're doing. And so at the same time, we're also developing patient-derived models where we're taking patients' tumors and we're trying to grow them outside of them ex vivo to sort of study what drugs would work best in them after they're removed. And so we're doing that as well. And so the big picture is we have these window of opportunity trials where we could actually treat patients and then take part of their tumors and see in real time, instead of waiting that time that I talked about with that vignette about my mom, is seeing in real time how those patients could respond based on looking at their different tumors.
That's awesome. That's awesome.
So,
That is, yeah.
and then here is just to show you that we really care about clinician scientists such as Dr. Shepard. So here are two examples, Dr. Isle and Dr. Worth, who are surgeon scientists. And so we really believe that these people, like Dr. Shepard, and having a kind of a holistic view of this disease is really going to make a difference. So let's skip over that. And then these are the people doing the work. And this was our group all together around Christmas time in the various labs at the Brendan Colson Center. So I will.
Thank you so much for all of that and for your commitment to doing this work, because those of us who have had it or who haven't now just so appreciate your willingness to talk to us and your willingness to continue the work to find something to help us have a better quality of life for longer. One of the things that always comes up, especially because some of us have children, is the early detection aspect of it. Has anything new come out in early detection other than scanning our children forever? And
Yeah. Well, so I think similar to the KRAS inhibitor work, I think there's a lot of excitement in this. There's a program that Dr. Sears and our group is involved in. It's called Pre-Seed, where they're basically collecting all these patients' samples who might be high risk, not, and basically being able to monitor them and try to figure out what's the best way to find an early detection signal. We, in Cedar at the night, we have an early detection program, and there's a lot of people, including the PCDC grant, which Dr. Shepard, Dr. Sears are PIs on, where we're basically trying to capture high-risk individuals, do kind of sophisticated imaging work with Alex Gumieres and others, and with our GI group, Dr. Cote, to really monitor these folks. There's our colleagues at Hopkins have a really interesting study that they basically have monitored high-risk individuals who come from high-risk families or might have a mutation. And they found that they're actually able to maintain clinically some of these patients to, live 10 years, beyond, presumably because of that early detection. So, these are expensive assays. And to your point, Stephanie, what we need to do is we need to find, maybe something better than it, but like, there's the PSA for prostate. Well, we need our sort of PSA that's going to be more specific. And we're doing a lot of work to get there. There's extracellular vesicles, there's other things, but, we'll get there.
And the other question I had, thank you for that, is about prevention. So is anyone taking these high-risk individuals and saying, well, is there anything we can do to prevent them from developing into cancer?
Yeah, that is something I'm passionate about with Dr. Shannon, and I think that's the whole point of maybe HOP and Healthy Oregon Project and POP, if you will, that we can identify these high-risk individuals. We could do interception strategies. Can we modify people's lifestyle? Can we monitor them as part of the high-risk group? This is certainly something that I think we could move the needle on and do better on. And it's something that I'm passionate about doing, because I do think that is a way to intervene.
Yeah.
But the best intervention is actually early detection
Right. At this
and
point.
then getting surgery
Yeah.
or early treatment.
The PanCAN has a program where they're telling people, to eat a healthy diet, to stop smoking, exercise, do all of those things. And for instance, we know that telling people to stop smoking prevents lung cancer. Telling people to use sunscreen when they go outside will help prevent skin cancer. But what I don't think I've seen any data to say, well, we prevented X number of cases of skin cancer because all these people used sunscreen, because you'll never know how many people you prevented it in, because or how many people are actually doing something preventative. So I always think that's an important thing, important message to get out there, that there are healthy lifestyle choices that people can make that may help. And it might not do anything huge, but a little bit is good, right?
Absolutely. I mean, to your point, the new data is emerging even about alcohol.
Mm-hmm.
Like, I don't know. I mean, I always thought, based on it's like, 1 or 2 drinks is fine. Now the data is emerging that it's really 0 drinks. Maybe unfortunately, but I mean, I think to your point, like, these lifestyle things are really coming out, that you have these large retrospective cohort studies that are really showing that no drinking versus never smoking versus stop smoking versus all those things really will move the needle and make a difference on your risk. I mean, it's the same data about statins with, heart disease, right? I mean, doesn't mean that if you're on a statin that you're not going to get heart disease, but you certainly can do a job of sort of preventing it, right? So I think these strategies are critical.
Thank you. Thank you so much for spending this much time with us, Dr. Brody. And it's been good to catch up with you, and I look forward to seeing you in April. Does anyone have any questions for Dr. Brody before we let him go back to his evening with his cute little boy?